Oh, we have to do two because
Yeah.
of the live stream.
Yeah. I'll tell him that too.
Do you want to just
I'll
speak
just hold it
normally
right.
and then I can give that to him to get started? Or do you want to talk through it too?
I'll talk. It's okay. You have to come up and do it anyways. It's okay. Good morning, everyone. Welcome. Welcome to the 12th annual ICCI conference. And there's two microphones here, not because I'm in stereo, but because we're recording this, live streaming it to YouTube, as well as recording it for later. Not all the talks, but certain ones that people have agreed to. A few little housekeeping things. There's another group in the cafeteria, so we're supposed to keep to the right side. So the wrong side would be the dairy group. Nothing wrong with them, but there's a whole dairy group in there, and we're on the poster side. So the whole cafeteria is taken up with conference today. So you're welcome to take your coffee and snacks and lunch even back in here if you need extra room. But you can also stand in there and admire the great posters, which would be good too. I'll just tell you that this is all possible through the Arthur Willis Visiting Professorship in Keynote Oncology and support from the OVC Dean's Office to run this thing with all the food and the posters and everything else. Anything else we have to say? I think that's it. We have the very, very last speaker couldn't make it today, so we have a little bit of extra buffer on time at the very end before our keynote, which is more time for coffee break or questions, of course, and discussion. Okay, so I'll let Michelle introduce our first speaker. So you'll have to put both mics on when you come up. It's a bit of a pain, but that's the way it goes.
Okay, wonderful. So our first speaker this morning is Dr. Jim Petrik. Jim is a professor in the Department of Biomedical Sciences and also adjunct faculty at McMaster and Western. And he leads a collaborative research program to discover novel therapies to treat advanced stage ovarian cancer. His longstanding collaboration with Dr. Jack Lawler from Harvard has resulted in a development of a mechanism to normalize tumor vasculature and enhance an uptake and efficiency of conventional therapies. Jim is extensively published and has been very successful in achieving amazing research grants, and he also sits on review panels for both CIHR and the Israeli Cancer Research Fund, and we're very excited to welcome him as our first guest this morning.
Awesome.
So,
Should get like
yeah, seriously.
for this.
Okay, that one.
Awesome. Thanks. Awesome. I feel like a cyborg now. This is good. I'd just like to start by thanking Jeff and Michelle for the invitation to come present some of our work and the rest of the ICCI committee. So we're really excited about sharing some of the work that we've been putting together for the past 15 years or so. So as most people know here, our lab's looking at ovarian cancer and some ways to address advanced stage ovarian cancer, which has some really particular challenges to it. So just by way of introduction, epithelial ovarian cancer is the most lethal gynecologic cancer. It has a very low 5-year survival rate, and its median age is around 65, so it's a postmenopausal condition. So there's some hormonal implications that we're looking at as well, but we'll talk a little bit about some of the vascular issues as a focus for our research. So EOC is a stage disease. So stage 1 disease is localized primarily to the ovaries. In stage 2, disease has migrated to the pelvic basin, but it's still contained within that area. At stage 3, it's metastasized now and spread transcolumically throughout the abdomen. And by stage 4, there's distant metastases. The numbers under here are the predicted 5-year survival rate if the woman was diagnosed at that stage. So if a woman's diagnosed at stage 1, it's almost a curable disease. The unfortunate thing is most women are not diagnosed until stage 3 because of the very vague, nondescript clinical symptoms that go along with ovarian cancer. So at this stage, there's typically a very advanced miliary disease, metastatic disease throughout the abdomen. So there'll be surgical resection of the primary tumor and then typically high-dose chemotherapy with platinum and taxanes. The challenge for ovarian cancer is that there's been no real therapeutic advancements and no real change in outcomes in 40 years. So we've taken on the challenge of looking at advanced stage ovarian cancer. That's the current clinical picture. So we're interested in looking at other mechanisms, other therapeutic interventions that we can develop that can help improve these outcomes. One of the things that our lab's really interested in looking at is tumor angiogenesis, which is the formation of new vessels from pre-existing vasculature. Any tumor that grows beyond 2 millimeters cubed is required to recruit its own vascular supply. And this is done by changing the balance between promoters of angiogenesis and inhibitors of angiogenesis. And I don't have enough time to go into a lot of the mechanisms, but some of the important promoters, vascular endothelial growth factor, which are referred to as VEGF, and thrombospondin is the inhibitor. This is the mechanism, the tool that we've been focusing on for the past decade or so, is optimizing ways to use TSP1 mediated approaches to help reduce this angiogenic drive. So as I mentioned before, a small tumor can exist happily and receive everything it needs through passive diffusion and osmosis. But in order for it to grow to become a more clinically relevant situation, it needs to recruit its own vascular supply. And it does that by very potently secreting pro-angiogenic factors. We talked about VEGF as one. There's a whole host of other ones. And so it floods the area with these pro-angiogenic factors. Adjacent vasculature will respond to this and start to sprout and mobilize vascular tubes towards the chemotactically towards the tumor that was secreting the pro-angiogenic factors. But the problem is the tumor doesn't really know how to regulate the process. It says, I need a vascular supply, so it really aggressively floods the system with pro-angiogenic factors. The vascular supply responds to that in a very aggressive manner because of the level of pro-angiogenic stimuli that's in place. So the result is that although there's a tremendous number of blood vessels that form in the tumor, they're actually very poorly organized, they're very poorly formed. So we end up with a very dysfunctional, disarticulated vascular supply that ultimately ends up, ironically, in resulting in a very poor perfusion of the tumor. So although there's tons of blood vessels there, they're not matured. So they don't actually carry much vascular supply to the tumor. So we end up with a situation. So if we look at a tumor, we end up with a very disorganized, dysregulated vascular supply. So we end up with a lot of necrosis, a lot of hypoxic regions within the tumor. And this has a problem from a number of different reasons. First off, we reduce supply to the tumor because of the vascular supply being so dysfunctional. We get very little vascular supply to the tumor. Because the tumor is growing and we have cellular proliferation and increase in size, we get increased pressure within the tumor. So increased interstitial fluid pressure collapses lymphatic vessels that reside within the tumor. So now we're not getting fluid in, and we can't drain the tumor because the lymphatic vessels are being collapsed and compromised. So we have a real challenge in getting things into the tumor, our therapies into the tumor. And I'd liken this tumor to my teenage boy's brain. So, their brains are already completely filled with knowledge and wisdom and everything, so it's jam-packed, so we can't get any more information into the tumor or into their brains. Same thing happens with the tumor. Okay, there's a tremendous amount of pressure, really reduced delivery opportunities, so it's really a significant challenge to get anything into the tumor. So as Michelle mentioned in the intro, I've been working for a long time with Dr. Lawler from Harvard, and he was the one that cloned thrombospondin 1 back in the day. Thrombospondin 1 is an amazing peptide, large protein. I can't really get into its biology, but it does a lot of really intriguing things. But the problem is it's a really massive protein. It's a 450 kilodalton glycoprotein. So it's not really a good therapeutic tool. So what we've done is we've isolated the type 1 repeat region out of the thrombospondin protein, I'll call it 3-TSR, and it does all of the awesome things that we want TSP1 to do from an anti-angiogenic perspective. It binds to its receptor, CD36, it has glycosaminoglycan binding domains, activates TGF-beta, so essentially it inhibits a lot of the pro-angiogenic stimuli. So we initially hypothesized that 3-TSR will specifically target dysfunctional tumor vasculature and in this result in this vascular normalization to reduce the presence of those dysfunctional, non-functional vessels in the tumor. So initially, in order to study this advanced stage ovarian cancer, we had to develop an animal model to replicate it. So it's a simple model where we orthotopically inject 1 million spontaneous. Those cells then can interact with the ovarian microenvironment. And we published a couple of papers showing that interaction with the microenvironment is actually critical in normal disease progression. These cells are p53 wild type. When we inject them, the cells that metastasize into the abdomen have developed a p53 mutation, which actually really closely replicates what happens in women. So the women, the cells derived from the ascites fluid in metastatic disease are p53 mutant, and we've been able to replicate that. So our disease model looks like this. We end up with animals that have large abdomen due to ascites or fluid accumulation within the abdominal space. They have large primary tumors, and then they develop metastatic disease through transcolonic metastases. The tumors are high-grade serous adenocarcinoma, which replicates about 90% of the epithelial ovarian cancer. So we've developed a model that has been a really good workhorse for us. It replicates the disease. We've staged it. We can allow animals to get to different stages and initiate treatment. So we've had a lot of work done with this model. So initially, we just wanted to see, does 3-TSR have any efficacy in tumors? So we initiated tumor formation. We started treating with 3-TSR, and we found that if we look at this tunnel assay, the green dots are the dying tumor cells. When we co-localized with CD31, which is indicative of endothelial cells, we saw that there was specific targeting of endothelial cells as well. So we saw that we could increase apoptosis both in the tumor cells, and we could reduce blood vessel density following 3-TSR treatment. So there seemed to be a fairly potent anti-tumor and anti-angiogenic effect. And I'll come back to that because we think that that's part of the impact of this therapy is it has a bimodal effect. It's cytotoxic to tumor cells, but it also has functions within the vasculature as well. So if we look, this immunofluorescence staining is looking at CD31, which is staining blood vessels in red. The green stain is for alpha smooth muscle actin, which is staining the smooth muscle support cells that line mature blood vessels. So blood vessels, the last stage of angiogenesis is actually maturation, where the tubes have formed, but very importantly then, we need to have smooth muscle cells that line and support those structures. And what that does is it provides infrastructural support, also provides cell communication with endothelial cells, which conveys a survival signal, but it ends up really regulating morphology and function of the blood vessels. So if you look in the top, the control treated mice that just received PBS, they have extremely large, really awkwardly organized blood vessels, okay? Whereas in 3-TSR, we've gotten rid of those really large dysfunctional tumor vessels, and we end up with very small, luminal, kind of appropriately formed or appropriately shaped vessels. So we've normalized the vascular morphology. And then when we look at tumor hypoxia, we've reduced the level of hypoxia in the tumor. So by inference, then we've enhanced perfusion to the tumors by normalizing and remodeling the vasculature. Which I'm not going to lie, when I say the hypothesis, where we said that we were going to preferentially target abnormal tumor vasculature, that was not our original hypothesis. I was trying to make myself look smart, that we actually predicted this was going to happen. This is a totally unanticipated outcome. We thought we were going to destroy blood vessels and starve the tumor in a traditional anti-angiogenic approach. At the end of the day, what we did is we shrunk the tumors down, we made them small, but we gave them an amazing blood supply. And initially we thought, this sucks, right? Like we're giving the tumor everything it wants, But what we found out is that, if we enhance delivery to the tumor, we already talked about before that delivery to the tumor is one of the major impediments to therapeutic efficacy. So if we can't get stuff into the tumor, how is it going to have its effect? So then we thought, well, is there any way that we can exploit this improved perfusion to enhance drug delivery? And we did a simple experiment where we just radio-labeled a chemotherapy drug. We injected it, and we saw that after vascular remodeling, we saw a significant increase in the radioactivity of the tumors, so a lot more of the drug got into the tumor. So this was our first kind of inkling that we may have an opportunity to enhance delivery to these tumors. Which is awesome, but completely clinically irrelevant. We need to look at advanced stage disease. Like I said, that's the clinical picture. So starting treatment at the initiation of tumor development isn't relevant. So we developed a model where we allowed mice to get sick and replicate about stage 3 disease. So they had large primary tumors, they had metastatic disease, they had the initial formation of ascites, and that's when we started treatment, because that's really the clinical situation. So what we did was we allowed them to go to about day 80, they had stage 3 disease, and then we started treatment with 3-TSR. And this was a really exciting result for us a little while ago now, but we saw that in the control group, this is what the tumors looked like when we started treatment. In the control group, they grew from this to this. But in the 3-TSR treated group, not only did they not grow to this size, but they actually shrunk. So this was an indication that with a 3-TSR, just as a monotherapy, it was able to induce regression of advanced stage disease. So we were pretty excited about those data. So then we wanted to look at, if we can optimize delivery to the tumor, can we now use this in combination? Can we use 3-TSR to normalize the vasculature to optimize or increase the efficacy or uptake of chemotherapy drugs? Most women and most individuals undergoing cancer therapy, regardless of what solid tumor type they have, undergo what's known as maximum tolerated dose, which means they get an extremely high level of delivery of the chemotherapy drug. They're given a drug-free interval to recover and recuperate, and then they're given subsequent dosing. For women, most women undergo a 6 rounds initial MTD of carboplatin paclitaxel. Extremely devastating on the individual from side effect perspective. There's a more recent work being done on metronomic chemotherapy, and there's a number of comparative oncologists here that are looking at metronomic chemotherapy in companion animal models, where we give smaller dosages, smaller dosages, but there's a chronic administration frequency. So we never allow the individuals to exist in this drug-free environment. The problem with this drug-free environment is this is where disease resistance occurs, or therapy resistance occurs. So in the initial administration of high dose, we may have cytotoxic death of a lot of the tumor cells, but the residual ones, that's a time where they can develop their resistance mechanisms. So drug efflux pumps and a whole host of other mechanisms to avoid the next onslaughts of chemotherapy administration. Metronomic chemotherapy reduces the problem of having the drug-free intervals. But the problem with metronomic chemotherapy is if you give really small doses, how are you going to get enough drug into the tumor? That's the reason why they have to give maximum tolerated doses. You give a whole crap load of chemotherapy drug in the hopes that some's going to get it into the tumor and have the cytotoxic effects that you're looking for. So we thought, look, if we've enhanced or increased efficiency of delivery, maybe we can optimize and allow enough chemotherapy drug to get in using the metronomic approach. So what we did is a simple experiment is we utilized 3-TSR on its own, maximum tolerated dose chemo, and metronomic chemotherapy alone or in combination with 3-TSR. A complicated slide, but what I'd like to just draw your attention to is that 3-TSR, when we've allowed the molecule to induce that vascular normalization, combine it with low-dose metronomic chemotherapy, it had the most potent anti-tumor effect, which it also had a very potent antivascular effect, reduced the microvessel density. But then when we did survival studies, when we looked at the controls, all the control mice were dead by 100 days. Chemotherapy drugs on their own improved survival, but then anytime we added 3-TSR, we saw that we had a fairly significant increase in survival. And then the group with the 3-TSR and metronomic chemotherapy, out to 150 days, which is half of a mouse's lifespan in some cases, we'd only lost 1 mouse due to disease. So we were really encouraged that this combination of vascular normalization and this low-dose metronomic chemotherapy had some really interesting outcomes. So the main findings for that is that, when we did the vascular normalization, we really increased the efficacy of these outcomes. So then we asked, could 3-TSR be combined with other treatment approaches? And this stimulated the collaboration with Sarah Wooten and Byron Bridle from pathobiology, where we looked at oncolytic viral therapy. So oncolytic viruses, and I'm not going to go into this too in depth, first, mostly because I'm not capable, but oncolytic viruses have a preferential cytotoxic effect on tumor cells. Normal cells typically are impervious to oncolytic viruses, where tumor cells are susceptible. But the problem with oncolytic viruses is that almost universally they cause a vascular shutdown. So you can get some oncolytic virus into the tumor, but then they cause a vascular collapse, which is a real problem, because if you want any subsequent therapy uptake, if you want to allow immune cells to get into the tumor after oncolytic viral stimulation, that's a real challenge because of this vascular shutdown. So we want to see if 3-TSR is really good at normalizing the vasculature and maintaining patent delivery to the tumor, can we overcome that vascular shutdown that's induced by the oncolytic virus? So what we did is an experiment. The oncolytic virus that we were using is Newcastle disease virus. So 3-TSR on its own, once again, shrunk the tumors. And this was done in the advanced stage again. So we allowed the mice to get to about stage 3. So we induced regression. Newcastle disease virus on its own also induced regression, but then the combination very potently regressed advanced stage disease. This arrow here, what I'd like to draw your attention to is if you look at this tumor, it's really blanched. It's an incredibly pale tumor, which really highlights and supports this theory that the oncolytic virus on its own causes this vascular shutdown and reduces vascular supply to the tumor. So, and if you look here in the combination, we can see that it apparently, in a gross perspective, is a reperfused tumor. So we've been able to combat that vascular shutdown, reestablish perfusion. At the same time, we're still inducing regression. Okay, so it still has a cytotoxic effect while allowing vascular supply to the tumor. So this is, in blue is hypoxia, in red is endothelial cells, and in green are the smooth muscle cells. So if we look, there's a lot of hypoxia in the PBS treated tumors. 3-TSR induces this vascular normalization. NDV, you can see rampant hypoxia, very limited vascular supply to the tumor, and a complete absence of mature blood vessels. So this is really kind of indicative histologically of that vascular shutdown that we hypothesized was going to happen with the oncolytic virus. And then when we combine, we see nice small luminal blood vessels, significant reduction in the level of hypoxia. So and once again, these tumors are really small. So we shrunk them down. We gave them good blood supply, and we dramatically increased the amount of oncolytic virus that was able to get into the tumor. But one of the main functions of oncolytic viruses, when you look from an anti-tumor perspective, is they have a direct cytotoxic effect. But one of the main effects that they have is that they stimulate intratumoral immunity. So they increase recruitment and activation of NK cells, of cytotoxic T cells. So one of their main jobs is to go in and start inducing lethal cytotoxicity, but also to stimulate that immune system so that you can have intratumoral immunity. But the problem is, if you recruit the immune cells, you need to be able to get them into the tumor. And there's some really cool pictures out there that following oncolytic viral therapy and that vascular shutdown, you can see immune cells lining the periphery and the perimeter of the tumor because they can't get in. So they've been activated and recruited, but they can't get access to the inside of the tumor. So this is a really incredibly complex slide, but what I want to highlight is that PBS, 3-TSR, oncolytic virus on its own, and the combination, when we look at intratumoral presence of these immune cells, so macrophages, natural killer cells, cytotoxic, CD4, CD8 positive T cells, we can see that in combination, when we normalize the vasculature, let more oncolytic virus get in, and then maintain that highway for the immune cells to get into the tumor, we can really increase the number of immune cells that get into the tumor. Okay, and we have some other data to go along with this showing the efficacy and the survival. These mice survive for a long, long time after we've done the combination therapy. But now what I want to talk about for the last couple minutes is where we're going next. So 3-TSR is awesome, but the problem with it is it's a small peptide and it's cleared from circulation in about 8 hours, in about 14 hours, sorry. So that's all right. It definitely has efficacy and has some biological function, but the problem is women are not going to want to go in for daily intraperitoneal administration of a therapy. So we've looked at developing a novel compound where we combine 2,3-TSR peptides with a non-immunostimulatory human immunoglobulin. And what that does is it increases the half-life from 14 hours to 8 days. So now this could be a weekly administration of a therapy, which is far more clinically relevant. And the other thing is we've seen FC3-TSR, which is the nomenclature for this fusion protein. So FC3-TSR also has increased biological activity. And it's through this concept of receptor clustering, where if you put two of the 3-TSR molecules, and we've seen this for other FC peptides, you get a clustering of the receptor. So you get increased biological responsiveness in addition to the prolonged half-life. So we're increasing time in circulation of a drug that does better things that we've seen with native 3-TSR. And we just did some initial experiments looking at the tumor outcomes. So if we look, we did different dosages, and the mid dose of FC3-TSR significantly reduces tumor weight and metastases, importantly, compared to 3-TSR on its own. So we saw 3-TSR did really fantastic things, but it looks like the fusion protein is even more potent. So it has more in vivo potency. We've shown a whole bunch of different in vitro efficacy that's better in FC3-TSR than 3-TSR on its own. And we looked, it's better at remodeling the vasculature, so we see an increased proportion of these mature pericyte-covered or smooth muscle-covered vessels in the FC3-TSR treated group. And then when we look at drug uptake, once again, we just radio-labeled chemotherapy drug, we see that FC3-TSR fairly significantly increases uptake of the drugs, even in comparison to 3-TSR, which we showed had significant in vivo efficacy. So lastly, one of the things that I talked about initially was that the increased interstitial fluid pressure collapses those blood vessels, lymphatic vessels, so we can't drain the tumor. So that's important from a fluid drainage perspective to reduce the pressure, but also the intratumoral immunity, if we increase the uptake of or activation of checkpoint inhibitors, anti-CTLA4, anti-PD-1, PD-L1, all of those things need to get to draining lymph from the data today, but we've been able to kind of reanimate the lymphatic function in these tumors. So if we look we're just looking at a stain that stains lymphatic endothelium in the tumors. In PBS treated groups, which has really reduced vascular supply, okay, you can see reduced vascular supply in the tumors. We have lots of hypoxia, and we have reduced presence of intratumoral lymphatic vessels. Whereas in FC3-TSR treated in the green, you can see there's lots of vessels that are still contained within the tumor core, which facilitates drainage, but also once we activate the intratumoral immunity, then we can now allow for those checkpoint inhibitors to drain to the lymph nodes where they really need to get to. So the working hypothesis right now, just in conclusion, is that an untreated tumor has really big, large, nasty, dysfunctional vessels, lots of hypoxia. We have collapsed lymphatic vessels, which really reduces the ability to populate the draining lymph nodes with immune regulatory molecules. Okay? When we treat with FC3-TSR, we reduce the presence of these dysfunctional vessels. We end up with small, luminal, smooth muscle cell covered functional vessels. We reanimate the presence of lymphatic vessels. We allow the uptake and activation of a whole host of different immune cells, NK cells, cytotoxic T lymphocytes, and then we facilitate draining of the relevant molecules to the draining lymph node, once again, kind of really kickstarting and amplifying the immune response. So that's where we're at right now. We're taking this, we're hoping to take this to human clinical trials within the next year or so. We've developed a small startup venture to attract venture capital to mobilize towards that. But the other thing that we're interested in doing is bringing this to companion animal clinical trials here at Guelph. I've talked about ovarian cancer, and ovarian cancer is the model that we've been looking at in the lab. But this is not restricted to ovarian cancer. All solid tumors that require vascular supply, which is basically all of them, should respond in a very similar fashion, especially other vascular hormonally responsive tumors. We're thinking of some of the soft tissue sarcomas, hemangiosarcomas, things like that. should respond very similarly to this. So with some of our awesome companion animal oncologists here, we're trying to establish some collaborations, doing some initial safety trials, and then move to companion animal clinical trials with this as well. Because the real strength of this is that any therapy that you need to get into the tumor, so chemotherapies, oncolytic virotherapy, immunotherapies, putting in FC3-TSR initially to normalize the vasculature should increase uptake and efficacy of those tumors. So we're excited to try those out in some of our hard to treat companion animal tumors as well. So the real take-home message of this is that drug delivery with this abnormal dysfunctional tumor vasculature is met with a whole host of different delivery problems. We can't get the therapy where it needs to go. Okay, we increase the efficacy and efficiency of delivery. That really should help increase the potency of current conventional therapies. And I'd just like to end by thanking everyone in the lab that's done a lot of work over the past more than decade on this. In particular, Kathy's been working on a lot of the FC3-TSR work, so she's done some really good work on that. I'd like to thank the collaborators here at Guelph, looking at the oncolytic virus and immunotherapy perspective. Great thanks to Jack, who's been a fantastic collaborator on this for the last long time, and then obviously funding. We really appreciate that. And thanks for your attention. I'd be happy to take any questions. Yes.
Jim, thanks for an excellent talk. Stupid plant biologist question, so I'll preface it with that. Just to ask, do you get any off-tumor effects? That is, 3-TSR has an effect on normal angiogenesis, so what happens in the rest of the body that's honestly, that's the important million-dollar question. And so what we found is that even in the most aggressive physiological angiogenesis, so wound healing, the angiogenic mechanisms build in that whole maturation as the blood vessels are developing. We found that FC3-TSR and 3-TSR on its own is the blood vessels that have smooth muscle cell coverage are impervious to it. So we've done wound healing assays where in our animal models, we've induced wounds and we've given them high dose FC3-TSR, and it does not impede normal angiogenic mechanisms. So the wounds heal at the identical rate. We've done retinal corneal vascularization assays. We see no impediment or impairment in normal angiogenic functions, which is important because a lot of our normal physiological processes, reproductive processes, especially in females, ovarian cycle, menstrual cycles are very dependent on normal vascular function. So we're really encouraged that we don't seem to be derailing that. Yes?
As far as your combination with metronomic chemotherapy, the doses, metronomic chemotherapy, the primary target is in the cancer cell anyway, so there must be something else going on that's causing the synergy that you're seeing.
Yeah, absolutely. And what we think is that, and we haven't looked at this, we're going to go back and do these experiments because we weren't thinking of it at the time as the immune response. So metronomic chemotherapy, I agree, is cytotoxic to the cancer cells, but Bob Kerbel's done a ton of work on the immune responsiveness to metronomic chemotherapy. It has some anti-angiogenic effects. We think that we're kind of trumping that a little bit with the FC3-TSR, but I think it's the immune response to metronomic chemotherapy that we're helping to accentuate. Brenda. Yeah.
And the interesting thing about ovarian cancer, as you pointed out, is that the cancer cells themselves, the neoplastic cells themselves, rely on VEGF signal. So I'm wondering if this dual effect is the real key reason why 3-STR works. And have you thought about looking at it in other models where there is neoplastic expression signaling, like melanoma, for instance?
Yeah. Right. So you're absolutely right. So the thing is, if you look at it from a just solely an anti-angiogenic perspective, it's kind of boring. I agree. Like you look at Avastin. Avastin has had not awesome effects, which targets the VEGF pathway. But I think the secret to why we see efficacy here is that it has completely non-VEGF related functions. So we take the VEGF side, we put it head to head with Avastin before, and it does way better job because of its specific effects that signal through the CD36 receptor. We found that any tumor cell, and we've done a bit of a panel, any tumor cell that expresses CD36, which is receptors, they're quite responsive to it. So we think that that's the key, is that tumors that express CD36, and we're going to do a panel of the companion animal tumors to identify expression. Most of the epithelial tumors are CD36 positive. We're just starting to branch out now. But I think that's the key, is if we divorce ourselves from just the specific VEGF-mediated effects, it still has a lot of efficacy mediated just specific through that signaling pathway through CD36. Jeff?
The ovarian model and the actual disease is a bit unique in that it's transcelomic
Yep.
metastasis.
Correct.
And some of these anti-angiogenic therapies have failed because they increase metastasis, but that's all hematogenic spread.
Yep.
I'm worried that that's gonna, like you found something that works, but it's working in a very unique situation.
Yep.
Do you think any evidence that it doesn't increase metastasis by?
No, it's a great question, right? The one thing we've done, it's still in ovarian cancer models, remove the primary tumor, and it still has efficacy in the metastatic disease. Where sometimes if you remove the primary tumor, metastatic disease just goes rampant. So still is able to put the brakes on that and induce regression in that system. But no, we haven't. We're just starting to branch out. There's been a lot of work just in this model. We're now starting to branch out to, we think it's, prostate cancer is going to be relevant. It's very similar to what we see in ovarian cancer. But no, distant metastatic disease, we can't replicate metastatic, like hematological metastatic disease, stage 4 disease in the mice because they die first, as most women do. So it's a great question, and we need to start working in other models. We just need some grant funding to do that, which is the bane of all of us.
Just to
Roger.
follow up on that, I was wondering, same sort of thing, most of your data is on the primary tumor. Do you see the same vascular effects on the
Yeah.
metastatic lesions, or are they too small?
Yeah, no, it's a great question. So we see identical things in metastatic. So all the if you can see the tumor, it's big enough that it needs its vascular supply. So we get some fairly large metastatic tumors, and they've completely developed their own vascular system, and we see the exact same thing. And Kathy's done a bunch of work where we've taken out the metastatic tumors, and we see increased uptake, increased delivery of immune cells, increased chemotherapy drug uptake in the metastatic tumors as well. So and I think that's why it's particularly effective in metastatic disease, which is really the critical thing. The primary tumor, although we use it as a model, it's really a surgeon's problem. No offense. But the metastatic disease is what ends up killing the patient. So we're more interested in does it have an effect on the metastatic disease? We use the primary tumor just to show biology. Alicia.
These are a quick mechanistic question. So your peptide is inhibiting, it's destroying the vasculature that is abnormal, or is
Yeah.
it also increasing the recruitment of pericytes?
No, it does. We don't think that we haven't really looked at, it's a good question because we haven't really looked at PDGF receptor and other things that stimulate parasite. What we do see is it induces a really rampant apoptotic cell death of those non-parasite covered. So we think it's completely
So do you know if the domain that is activating TGF-beta, which is included in that peptide,
yeah.
have any function on the peptide?
I don't know. To be honest, I'm resistant to get into TGF-beta because I'm not smart enough to, it's way too complicated. I hate it. Jack has an interest in TGF-beta and I don't. So we don't know what the TGF-beta mediated effects are doing. We've done some TGF-beta mutants where we've knocked out the TGF-beta activating domain in the 3TSR and it really reduces the efficacy. So we think that, we don't know why that is, but it seems like there's some TGF-beta mediated effects, but we haven't had a chance to pursue it.
So the TGF-beta promotes vessel stabilization.
Right. Yeah, absolutely.
That perhaps there is an increase in the recruitment of pericytes.
And we actually thought that by mutating it, we'd increased efficacy, but we did the opposite because TGF-beta never behaves as you think it should, right? So we don't know. We haven't pursued it.
I have time for one more question.
Yes.
I understand that it's really early on in the stages of the clinical application.
Yeah.
But any preliminary data on safety?
That's what we're just hoping to be able to do that initially in a healthy cohort of companion animal patients, just to do the safety data. I can tell you in our preclinical model, we've seen no indication of any cytotoxicity or off-target effects. So the animals that we give even very high doses to are still reproductively competent, which means that their vascular function is still maintained. We've done the wound healing assays. We see no other sequelae in our preclinical models. We want to do it in companion animal healthy cohort. which then, with those safety data in hand, then we can move to companion animal clinical trials and the human clinical trials as well. Awesome. Thank you.
Great. Our next speaker this morning is Thomas McLaughlin, and he is from the Department of Pathobiology. He's going to tell us a little bit more about some of the stuff that we've seen highlighted to start. Do you need your computer?
I'm just gonna, it's on here already.
Okay, perfect. Do you want to hook yourself up?
Yeah, how do I need to do it? Just put on the right one.
Either one is, yeah, just wherever.
Do I need both of them?
You need both.
It's not recording, I can use it.
This one, oh, did you ask for this one not to be recorded?
Yeah.
Okay, that's
I
fine
realized
then,
it
so
was not
just that one is fine.
This one?
Yep, no problem.
Hi everyone, my name is Thomas and I'm currently a PhD candidate in Dr. Wooten's lab here at the OVC. And what I'm here to talk to you today about is viral sensitizer mediated enhancement of oncolytic NDV leads to rapid